Okay, we're going to begin our discussion of metabolism by kind of getting the broad processes sorted out. As we move forward with this, you're going to want to have different uh, colors available to you. I'll be using 4 different colors here. Black for paths and parts. You'll see those as we generate templates. I'll use red for hormones where they're important in metabolism. We'll use green for the nutrients or the metabolites, and we'll use blue for the metabolic pathways and the processes. So, for example, I'll use G for glycolysis, P for protein prep, F for fat prep. You'll understand all that as we. So, we're thinking about a dairy cow. The ability to produce milk comes down to metabolism. So, I'm going to propose over the next couple weeks that a cow is metabolism. I'll try to prove that to you. So, here's basically metabolism. Cellular metabolism. Some of you may have seen parts of this before. Some of you haven't. This is we're taking from our anatomy and physiology of the farm animals. There's lots of different diagrams out there. We'll show you 2 different ones in this lecture. We generally have the things that happen in the cytoplasm and then the things that happen in the mitochondria, more or less. So we have a transition from one to the other. There's a lot of different names here: the Krebs citric acid cycle, TCA cycle, tricarboxylic acid cycle. Lots of different names for that. When I went through some of my classes, I had to memorize all the structures, what all these looked like, what they were, what they weren't. For a freshman level class, first year level class, I'm not going to ask you to do that. Those of you going on to grad school, it might be helpful to you to start doing that. But I think more important at this level is we understand what's happening. What are the consequences? What are the inputs, the outputs, as opposed to the details on the inside? It's easy to test the details, simple multiple choice. Is it right or is it wrong? But you really need to understand things. And as we get to the other side of metabolism, you'll understand why I did that. We're going to explain why ketosis happens, while milk fat depression happens. You're going to see through metabolism how all this stuff, mistakes occur, and we get these metabolic disorders that are related to metabolism. I'm going to start out with glycolysis. Glycolysis is pretty straightforward. We have glucose, which is a 6 carbon compound. We introduced those in the carbohydrate section. That's what goes into the glycolysis. What comes out is 2 pyruvates. Basically, you break the glucose molecule in half into 2 pyruvates. Along the way, that process generates a net 2 ATP, and it does it very quickly. So we need 2 ATP to prime the pump. We produce 4 at the end, so that's where we get our net of 2. There's no oxygen required for glycolysis, and it all happens in the cytoplasm or the cytosol. So very quick process when you're sprinting. This is probably where you get most of your ATP from. You're not really burning things. It requires a little bit more effort, a little bit more time. The initial thing there would be glycolysis. In our diagram, glycolysis is in this section right here. We start with our carbohydrates. We break them down into 6 carbons. We got glucose, glucose phosphate. We add those ATP in, and at the other end, we get pyruvate or pyruvic acid out, a 3 carbon compound. Process we'll talk about is oxidation. Oxidation takes 2 carbon units, basically molecules that have two carbons. Doesn't really matter so much whatever's attached to them, but two carbon units, and they burn them completely to CO2 and water. So the carbon units that go in are pretty much completely combusted, so we get the CO2 and water. We generate about 34 ATP per glucose. So we'll show you here that for every glucose, we have two oxidation cycles, two carbon units that go into oxidation. Each of them gives us 17 or so ATP, more or less, depending on where we are, depending on various efficiencies. We might have anywhere from 32 to 38 per glucose unit. In order to burn completely, we need air, we need oxygen specifically to let that happen. This happens in the mitochondria, and you're going to hear it called a number of different things: TCA cycle, tricarboxylic acid cycle, the Krebs citric acid cycle, the citric acid cycle. All those things are the same. Two carbon units to CO2 and water. So we look down here, this big circle here, the cycle is oxidation, and we burn with oxygen to release all the energy remaining in the parts that get this far. Next thing we'll talk about is fat prep. We've talked about how carbohydrates come in. Now we have to talk about how a triglyceride comes in. So the first thing that happens in our fat prep or prep fat is the lipolysis. We split the triglyceride into its parts. The glycerol, and this feeds directly into glycolysis or oxidation, depending on where things are. And then we have three fatty acids of varying chain lengths. Then we need to take those fatty acids and we will need to break them down into two carbon units. So chopping off two carbons at a time, which are fed directly into oxidation. All this is going to happen in the cytosol. At our diagram of cellular metabolism, you can see that the fat prep is over here. The fats over here is triglyceride. We're breaking them down into fatty acids and glycerol. Glycerol is going to feed into the glycolysis pathway. The fatty acids are going to be broken down by beta oxidation into two carbon units. The acetic acid has two carbons in it, little modifications, and they are fed directly into the carboxylic or the TCA cycle. So glycerol goes this way. Our fatty acids broken down, two carbon units, they get fed directly into the TCA. So fat prep is over here. Prep and protein. This is a little bit more complicated just because of the variety of amino acids and what gets broken down. We saw those R groups. They vary quite over a wide span from no carbons to 20 or 30 carbons as an R chain hanging off. So first thing with a protein, we use hydrolysis reaction to split the protein into its parts. That's going to yield amino acids. We take that amino acid, the carbon-carbon nitrogen unit, and then we rip off the ammonia group, which is to say it's deaminated. So we take everything, which is usually hydrocarbons mostly, and we separate that from the ammonia group that is at one end of the molecule. The hydrocarbon is then prepped and adjusted, and it's going to fit into glycolysis or oxidation where it fits best. So we may be able to break it up into pieces and it goes directly to oxidation, or you've got some 3 carbon units or 6 carbon units that are going to go into glycolysis, wherever it fits into the system. As a byproduct of this protein prep, 2 ammonias are stuck together to form urea, which is either recycled in our ruminants, or it could be recycled a little bit in the horses as well, or excreted as a waste product out the kidneys. And again, All this happens in the cytoplasm or the cytosol. So protein prep over here, we're taking whole proteins, we break them down into amino acids, we take the ammonia off, that goes that way, and then all the parts are going to feed in where is most appropriate for a piece of that size. So either into glycolysis right here or directly into the TCA cycle. Transition. If we look at transition, we take 3 carbon units that come out of glycolysis and we reduce them to 2 carbon units and carbon dioxide. We're going to say in this class that ATP is not generally generated. So we just kind of pull that carbon off and we're left with 2. And that 2 carbon unit is put into oxidation to be burned to CO2 and water. So I call it transition. I don't know if anybody else does, but that's the way I remember it. You got your transition is going to happen at the end of glycolysis. You got pyruvic acid. You're going to lose the CO2, and you're going to generate this 2 carbon unit in some way, shape, or form. 2 carbons go in. We have oxaloacetic acid. 4 carbons, 2 plus 4 gives you 6. We have 1 CO2 come off. 2 CO2s come off. So the 2 that came in have been burned off. And then we run back to the beginning with our 4 carbon unit to pick up the next 2 carbon unit. I like to think of this sort of as a roller coaster that's gone bad. We have our cart here. We pick up our 2 carbon unit. They get flung off here and then here. And then the cart returns to the beginning of the line empty to pick up the next 2. Maybe that's not the best way to think about it, but that's how I remember it. I'm kind of weird that way. So we see all the parts kind of hooked together in some way, shape, or form. We need to think about the flow. How do things work together? How do things happen? So if we take a carbohydrate here, glycolysis, we break it down, it's going to go through, glucose is going to go through glycolysis, transition, oxidation to be burned completely to water. Protein prep, you're going to either have proteins, they're going to be prepped first, they're going to be fed into glycolysis, transition, oxidation, or protein prep, transition, oxidation, or protein oxidation, depending on the size of the deaminated hydrocarbon backbone. Protein or fat prep, you're going to have fat prep. Glycerol is going to go into glycolysis, transition, oxidation. The fatty acids are going to do fat prep and go directly into oxidation. So another illustration of doing the same things. This is a book out of one of the textbooks I've looked at. We're going to do red are catabolic reactions. We're breaking things down. Blue are anabolic in these diagrams, building things up. You're going to see the same format here. We've reversed the fats and the proteins from our other diagram, but you can see the things that are happening. You've got glucose, the monosaccharides fed through glycolysis. We get pyruvic acid out. You've got fats being broken down into fatty acids and glycerol. Glycerol feeds into glycolysis. The fatty acids are going to skip down to oxidation. Amino acids, depending on how big they are, we're going to break them apart. We're going to deaminate them. The ammonia goes to urea, and then the amino acids can either feed into glycolysis or transition or directly, as in the next slide we'll see. So you've got oxidation directly in. You've got things feeding in your 2 carbon units. hitching the ride going around. You've got CO2 coming out. You've got the high energy units that are used to generate ATP, oxygen. We reduce CO2. Oxygen comes in, CO2 comes out, water comes out. So the Krebs cycle is going to generate those high energy units. Ammonia goes to urea, goes to urine. So this is another diagram, a little bit more simplistic, but illustrating how all the patterns fit together. We'll call this central issue here the transition period, actually from here to here. 3-carbon unit to a 2-carbon unit, and we feed those in, the carbons come out. So we have our ATP being generated, and then we have work, and then we break it down to ADP plus an inorganic phosphate. We generate the reducing equivalents to generate those again, and the cycle goes around. So blue we're building up, red we're breaking down. So we've got our 5 processes. We got to get to know those. Forward, this is how I'll abbreviate them on our charts and diagrams. So glucose, CO2 and water, GTO. Fat to CO2 and water, we're going to have part of it's going to go FGTO. You got some of it's going to go FO. Protein, depending on what amino acid it is either going to go PGTO, PTO, or PO. All those processes, pathways will take an organic compound, fat, carbohydrate, or protein, and turn it into CO2 and water, generating energy along the way. If we go through and summarize what we've talked about so far. So we have each of our processes here, the code letter that we're going to use in this class. Very simply, what happens? Glycolysis, we have 6 carbons going to 3 carbons. No oxygen required, 2 ATP generated. Where does it happen? The cytoplasm. Prep protein to amino acids, to ammonia and a carbon skeleton. We don't require oxygen for this. There's no ATP generated. We have things prepped in the cytoplasm, so they might go on to be respired, burned, to CO2 and water. Prep fat, triglycerides into the 3-carbon, which is glycerol, and then 2-carbon units. We take the fatty acids, chop off 2 carbons at a time. No oxygen is required, happens in the cytoplasm. We'll abbreviate that as F. Transition, wherever you got those 3-carbon units, we got to break them down to 2-carbon units. Loses carbon dioxide. No oxygen is required. There may or may not be ATP generated. We're going to say no for the most part in this class. Cytoplasm, oxidation, 2-carbon units burned to CO2 and water, very much requires oxygen. You notice that's different from everything above. Generates a lot of ATP. That's pretty different from everything above. Happens in the mitochondria. Again, different from everything above. So comparing and contrasting, there's a lot more similarities with these 4 processes than there is with this. So the cow is metabolism. This is the basis of the processes that we're going to use. We're going to talk about where these processes dominate and where they're less important, how the liver functions, how fat tissue functions, how muscle, brain, what's the other one? Muscle, brain, and the mammary gland function. We'll make sure that you understand those in the coming week. I want you to think about how does glucose get converted to carbon dioxide and water in an animal cell. Line up the steps. What are the processes that have to happen for glucose to be turned into CO2 and water? So Which steps do we go through on this diagram? So I went through them. Make sure you can do that. Answer that question. Do the same thing with protein. How does protein get converted to carbon dioxide and water? Line up the steps. How does protein get there? And with protein, we have multiple pathways, so make sure you get them all. Triglyceride, we got multiple steps in the prep phase. How does that get converted to carbon dioxide and water in the animal cell? Line up those steps in order and know which goes where and why. Fat, where does the glycerol go? Where does the fatty acids go?